Welcome to our presentation on DNA. DNA replication occurs because cells divide, and if they didn't copy their DNA, then they would run out. DNA consists of two strands in a double helical shape, not pictured, that are antiparallel. Each strand consists of a sugar, a phosphate group, and a nitrogenous base, either adenine, thymine, cytosine, or guanine. When only one of these components bonds together, they create a nucleotide. The two strands are held together by hydrogen bonds, or H bonds, and in the first step of replication, helicase breaks apart the H bonds of the DNA like a zipper. The resemblance is uncanny. So as the helicase unzips the DNA, the two strands split and are called parent strands, and their bases can be used as templates for the new DNA. The bases of DNA undergo complementary base pairing with free-floating nucleotides, and DNA polymerase binds the phosphate and sugar groups together to create new strands of DNA. As you can see here, yellow with orange, pink with purple, or in real life, guanine with cytosine and thymine with adenine. After replication, the amount of DNA has doubled. Each strand consists of a mix of one parent strand and one new strand. Cool fact: DNA replication is semi-conservative, just like my parents! Which means that half of the DNA is saved, the parent strand, and the other half is new, the new strand. Now on to protein synthesis. While DNA can replicate, the real purpose of it is to create proteins. There are two steps in protein synthesis: transcription and translation. In transcription, RNA polymerase unzips one area of DNA, just like helicase. RNA polymerase also aids in the bonding of free-floating nucleotides together, just like DNA polymerase. mRNA is created from DNA with the same rules of complementary base pairing, except instead of pink, thymine, RNA uses green, uracil. Only one strand of DNA, the antisense strand, codes for mRNA. The other strand, the sense strand, just kinda sits there until transcription ends. Once the mRNA has been transcribed from the gene, the RNA polymerase falls off the DNA, and the base pairs sort of just come back together, like a zipper, because H bonds. The mRNA then exits the nucleus to the cytoplasm, awaiting translation. Now up to this point, we've only learned that bases are organized in long strings of letters. Warning, this next fact will surprise you. The bases are actually grouped into triplets called codons. In the cytoplasm, there exist lots of friends of the mRNA. tRNA, ribosomes, and amino acids are their best friends. Translation occurs when a ribosome bonds to mRNA, and tRNAs begin to bind their anticodons to codons through complementary base pairing. The amino acids attached to the tRNA link together and form peptide bonds. When the ribosome reaches a stop codon, everything falls off. Translation actually occurs very quickly, at 20 amino acids per second, so a protein with 200 amino acids would only take 10 seconds to be made. Ta-da! You just witnessed the birth of a protein. How beautiful.